I'm Bob Desimone. I'm the director of the McGovern Institute for Brain Research here at MIT. Just very briefly, the McGovern Institute was founded in the year 2000 by Pat and Lori McGovern with a dual mission to understand the mind and brain and then to apply that knowledge to helping people with many different forms of brain disorders, including Parkinson's disease. As part of our outreach, we periodically host events like this for our friends and supporters and members of the public who are coming in for the first time. And it is really a thrill for us to be able to talk to you about some of the things that we're doing here. And we'd love to get your feedback, and we'd love to have you stay involved with us and sign up for the newsletter and so on. We have a really exciting, I think, schedule planned for you. And I want to thank you for being here. Unfortunately, we have a government agency visiting us at exactly the same moment, and I have to make a pitch for money. So I hope you will excuse me because I need to run out for a bit, but I will be back, and I'm going to turn over as Master of Ceremonies to Charles Jennings, who's the director of the McGovern Institute Neurotechnology Program and our head of communications at the McGovern Institute. Charles himself has a long and distinguished career in science. He was a former editor at Nature, the premier science magazine, founded Nature Neuroscience magazine, worked at the Harvard Stem Cell Institute and the Allen Institute, and we are so fortunate to have him here working with us. And Charles will introduce our upcoming speakers and handle things while I grovel.
Great. Okay, thank you, Bob. And welcome, everybody. It's a great pleasure to see so many of you here, old friends, new friends, and we're really excited that you're able to join us. We're very grateful to the American Parkinson's Disease Association for the opportunity to partner with them in this event. As Bob mentioned briefly, this is one in a series of outreach events that we have been holding with different disease advocacy organizations, really to try to sort of spread the word, if you like, about the work that we're doing and the ties that we are building to the clinical research and clinical community and colleagues. So tonight's or this afternoon's theme is Parkinson's disease, and we have a really exciting lineup of speakers for you. Just briefly to tell you the format, we will have about probably not quite an hour of presentations from our three speakers, and we will then open the proceedings up to a question and answer session with the floor, and we hope that you will all pitch in and fire any question that you want at any of us. We really enjoy these to and fro's, and we hope you will too. We will then have a reception outside starting at 5:30, and for anyone who is interested, we'll also be providing a tour of our imaging center, so you can come and see some of our newest toys, the equipment that we use for scanning the human brain, and some of the most advanced brain imaging equipment in the world is right within this building, just a couple of floors below us, and we'd be delighted to let you take a look at it. So with that, I'll move to the speakers. So we have three speakers tonight. I will introduce them in turn, but we start with Roberta Sidney, who I think many of you know as the chair of our Friends of the McGovern Institute. Roberta will give an overview from a lay perspective, what it's like to be a family member, affected by brain disease. We will then move on to Anne Graybiel, who is a professor here at the McGovern Institute, who will give you a sense of the research that we are doing here on Parkinson's disease and on the neurobiology related to Parkinson's. And then we will, the final speaker will be Marie Saint-Hilaire, who is the director of the Parkinson's Disease Movement Disorder Center, and I'll come back and introduce her in more detail later on, but she will give a clinical perspective on where the field is at in terms of the development of new treatments. But I will start with Roberta. So Roberta, as I said, is the chair of the Friends of the McGovern Institute. She's a tremendous and dynamic supporter of our work, and she will tell you herself about how she became interested, but I wanted to add a particular welcome to Roberta's parents, Stanley and Sheila Sidney. We are so pleased that you were able to be with us. So thank you so much. And Roberta is the chair of, is the president of Sydney Associates. That's her day job. But as you will hear, she's also a tremendous advocate for brain research. So thank you, Roberta.
Okay. You going to show me what I press? Okay, good. Yeah, that's me. Which one? Okay, great. Okay, so I'm a real estate developer. That's what I really do. I'm not a Parkinson's expert, but I've become one. So I'm going to start with a little story. So first of all, anybody in the audience with any back problems? Yeah? Okay, good. So picture this. You're downsizing. You're moving from a big house in Chestnut Hill, and you're moving to a city apartment. Okay, picture that, right? So you've got to go through closets. You've got to climb through underneath counters and pull things out and reach here and reach there, and all of a sudden you do one last thing and you go, uh-oh, and your back goes, that's it, I can't move. And you're in spasm, you can't do anything, you can't walk, you can't sit up, you can't turn over. Well, that was me 3 weeks ago. And I tell you this story because I knew that I was going to get better. I mean, obviously, I'm standing here now. I couldn't even get to the bathroom on my own steam. It was pitiful, but I'm better. What isn't so easy is for someone who has Parkinson's disease. When they have an issue of mobility, it doesn't just get better in a few weeks or a few days. So I'm going to speak a little bit about what it's like to live with Parkinson's, try to speak from the perspective of someone who has lived with it, and give you a flavor, hopefully, of what that's about. So, click. Okay. So picture this. Here you are, young boy, playing the trumpet. Everything works well. Your hands work. Your mouth works. You're doing your thing, and you're thinking about one day, maybe you will get the grades and you will go to MIT. That's kind of all you're thinking about. You're not thinking about anything that's going on in your brain. You're not thinking about dopamine. You're not thinking about popping pills. You're not thinking about anything else. You're just having a really good time. So then, things look pretty good. You move forward. You actually get to MIT. You get your BS, your MS, you're a civil engineer. You go out on your own, you get this really big sign, let everybody know what you do, and you're on the job site, you're walking easily, you're doing everything you want to do. You rock out with your wife, picture. You go on safari, you're bending down, you're holding a gun, you're looking really cool, and nothing seems to be wrong. You're just doing what you're doing. You become a very young grandfather. Can everybody see the pictures? I guess they're having trouble, huh? Okay, that's the wrong way. I can tell from here that's definitely the wrong way. I don't want to touch anything. How are you doing? So this is my youngest sister on the left and my oldest niece on the right in Dad's arms. Okay? You have no idea what's coming ahead. You know, you're just blissful, having a good time. And just to point out how mobility is not a problem, this is right before the backflip. I know, pretty cool, right? So then Parkinson's hits. And it's not so easy anymore. You're still able to stand up, walk around, but your face has a mask. It's a little bit difficult. Smiling doesn't look quite the same, but you're out there, and these are all the grandchildren. And the one on the left in the pink, that is the one that was the little baby. She's now 31. But this was actually a few years ago, because the baby that's there is now 7. So you can do a little image. But this is a few years ago with Mom and Dad standing with all the grandchildren. But you persevere. You're still playing the trumpet, not quite so easy anymore, still playing chess. but it isn't quite the same thing. Then you have more limitations. You're in a wheelchair. You're not able to walk anymore. You're using a walker for therapy, but you're really kind of sedentary. But you're still going. This is the thing. Parkinson's doesn't stop you, and you still are going. This is now a wedding. So this is me and my brothers and sisters with Dad at a wedding. And Parkinson's has taken a lot of your ability to calculate arithmetic. It used to be so easy. When you were at MIT, you were so clever with math that the math professor said, I'm not going to give you the exam. Your exam is to create the exam for everyone else. The only requirement is I want you to be able to solve every problem you put in front of everyone. When Dad had his deep brain stimulation surgery 10 years ago, if any one of you knows about the surgery, you stay awake when they actually implant the electrodes, and they're doing different things with you and asking you to perform certain activities, open your hands, close your hands, move your feet. But they also ask you questions, you know, who's the president? Well, one of the things they ask is they ask you to count backwards by threes. Well, that was too easy for Dad, so they said count backwards by 7. I mean, I couldn't even do that now. I mean, so that's the kind of mind that he has. So what I would like to say is that I feel very, very fortunate to live in a community where Boston Medical Center exists, where Dr. St. Hilaire works. Dad gets treatment in that facility. It's the best place to be sick. I think if you have to be sick, this is one of the best places to be sick, the city of Boston. And I also feel extremely fortunate to be affiliated with MIT and the McGovern Institute, where people are working very hard with Dr. Graybiel and everyone in her lab to crack the code. Because for the trajectory to be different for people in this room, for people who come next, who are going to be diagnosed tomorrow, we need to know more about what causes the disease, what's going to ultimately turn into a therapeutic difference in terms of the lives of many people. So I'm excited. I'm looking forward to hearing from our next speakers about what they're doing and what they're learning. And the last picture I'll leave you with, because it's always nice to be upbeat, is still going strong. This is Mom and Dad having a good time. This is, I think, in New York a few months ago. So I just thought I'd give you a picture. It's down, but not out. So thank you.
Great. Well, thank you, Roberta, and thank you to your family for sharing your experiences with us. Very moving presentation. So our next speaker is my colleague Anne Graybiel. Anne is a professor here at the McGovern Institute. In fact, she is what is known at MIT as an institute professor, which is the highest academic honor that MIT confers on its faculty. So we were absolutely thrilled when Anne was awarded this honor. She's received many other honors in her time. She's a member of the National Academy of Sciences and a winner of the Presidential National Medal of Science, and is one of the most eminent researchers in the field of Parkinson's disease and basal ganglia. And Anne is going to talk to us about her work. So, Anne, welcome. Thank you. It's all true.
Well, that doesn't really sound much like me. But
And you know what? We forgot to say one thing, which is that you're good. I am so pleased that there are 3 women on this panel. Did you even notice that? 3 women. Is that amazing? Right? Right? I just have to say that because it's not always the case.
I'll contribute one word for
isn't she great? Really, Roberta. Thank you so much. And I'm so pleased to see your mom and dad in the back. And they're wonderful people. Your family is wonderful. And there's several people from our lab here and over there. And I can tell you that every day we are inspired by your story and by your support and by your enthusiasm. And I don't quite know how to tell you all the way properly, but for a basic scientist to even contemplate being able to help with this disorder is over the top. It's wonderful. And so we, and I mean we, these wonderful people over there and maybe elsewhere in the back, we're all working hard. So Roberta said it. I guess for those of us who don't have Parkinson's disease, it was pretty easy. In fact, probably didn't even think much about coming in this room and finding a place to sit and sitting down, maybe being polite and saying hello to someone beside you, even if you don't know them. But as you pointed out, for someone with advanced Parkinson's disease, even these very seemingly simple behaviors become great hurdles. For example, sitting down or standing up or speaking clearly. So how could that be? And I think everyone in the room knows something about dopamine. You mentioned it, that magical neurotransmitter in the brain that bit by bit, slowly usually, but inexorably, unfortunately, gradually is lost in the brain. And as that loss occurs, eventually one sign and then another of Parkinson's disease occurs. And the signs may be different. For some people, there may be motor problem. We're familiar with that. For other people, it may be more a kind of a psychological problem, a kind of problem with emotion, for example, being changing a little bit different person. So the one thing we all can agree on is that a terribly difficult thing for a Parkinson's patient to do is simply to decide to do something and do it. And this was magnificently put by James Parkinson, and I have his words down below. And what he said was, the core difficulty in Parkinson's disease is the ability to translate your will, what you want to do, into action. So the normal brain, is there a pointer? I should have thought, is there a pointer? I guess we're MIT Institute of Technology, but I'm not sure we have pointers. We do? I could use the cursor. Oh, you're so nice. Thank you very much. So the dopamine, interestingly, shockingly, we learned, is mainly in the deep middle part of the brain. The part shown here in pink, it's one on each side, that's called the striatum, and you're just seeing the tip of the iceberg. It gets much, much bigger in another slice. And this slice actually is a very famous brain scan, one of the earliest ones, where the levodopa was radioactively labeled with very brief radioactive label, briefly there, and so they could see where this dopamine is in the normal brain. So if you now look at a comparable image, these were given me by Professor Garnett in Canada. Now you see that that dopamine, that pink, is almost gone. This person had severe Parkinson's disease. But to us, as neuroscientists, this is a spectacular mystery, because we've all thought and known, and we still believe, that much of what we think and do depends on this outer rind of the brain, the neocortex on the outside. We think, we act, we sing, we do math with this part. So how could it be that a defect in like one neurotransmitter deep in the brain, how could that so profoundly affect a person? And so that's what I'd like to talk about. And that's the mystery we're trying to solve. So there's one part of the mystery that we got a grip on, the field does, and that's that, curiously, the deep brain, which are called the basal ganglia in toto, they have these massive highways that eventually lead up to the front end of the brain. Here's the motor cortex, famously called the premotor cortex, and here this prefrontal cortex, as far forward as you can get, where we seem to do a lot of our online memory work, our little working memory it's called, and we plan ahead with this part, and in general we organize stuff with this part. That's the main target, outflow target of the striatum in the basal ganglia. And so what we think in the field is going on is that here we have this neocortex shown here in this cartoon, and it turns out it itself talks down to this dopamine-rich part, the striatum. This is the pink part, and here you can see the fiber tract carrying dopamine up to the pink part. And you can also see in this cartoon that basically there's a great loop. This thinking cap we have projects fibers into the deep brain. That deep brain gets modulated by the dopamine coming up here. That dopamine is carrying fascinating signals. They're sort of predictive signals that help us to choose what to do next. And so all of these loops that are going round seem to have something to do, seem very much to have to do with selecting and being able to do the next thing and the next thing until you get a whole sequential behavior. And once you do that behavior a few times, you get what we would call a routine. Now, if the dopamine, you can see in this diagram, gradually goes away, and it actually goes away because these tracts that I'm showing you here in this cartoon, that tract dies back. And as it dies back, not enough dopamine gets here, and then this region is disordered, and the selection, the ability, for example, to say, I want now to pick up this bottle, that becomes impossible because of the disorder deep down. And so that's part of this amazing and to us very surprising mystery that the deep part of the brain could influence what we do through its effects on the big king of the brain, the neocortex. So that's good. This is all good. So in fact, there are a lot of things now in the Parkinson's field that are definitely good, hopeful, promising. For example, we roughly know the circuits. I just showed you one cartoon. And we're more and more beginning to learn about some causative or predisposing genes. It used to be thought Parkinson's wasn't genetic. Now it looks like some forms, maybe many forms, behind them lies some darn mutation somewhere in some gene. We know that often the dopamine loss is relatively slow. The dying back is slow in that tract. And what that means is, that's good, because there's lots of time for therapies to be developed. And even if you're dealing with right now, there is a period when therapies can help, leading among all of the therapies being, as I think you all know, the levodopa therapy, such as Cinemet, that is given to patients. Now, and I have just put this deep brain stimulation that you mentioned, that your dad had that surgery, and maybe in the discussion period, I would love to talk about that. I'm probably going to run out of time, but I'm happy to talk about it. What I do want to talk about is that despite all those good things, we've got some pretty major challenges in the research side of things. And the first thing, and it's almost hard to believe, so I have to ask you to believe me, we still do not know what dopamine does to those circuits. It's crazy. We've known about the dopamine loss for years and years and years, since the 1960s. And bit by bit, people have learned many exquisite things about the action of this little molecule in the brain and in the circuits. But if you ask something simple like, if I can't pick up the water bottle, what is it about no dopamine that makes me unable to do that? We can't give a very clear and crispy answer. We need that answer, because if we can figure out what the dopamine does, even if we can't prevent it from going away, we might be able to zoom in there with a new method and mimic what it does do. There is another thing which I'd like to talk about just at the end of the talk, and several people here, Jill Crittenden among them, has worked together with us in the lab very hard on this problem, which is, as some of you know, for someone who is given the levodopa therapy, after a certain number of years, there may be complications. So paradoxically and sadly, now the person who's sort of frozen, locked in, suddenly starts making movements that the person doesn't intend to do, famously in the case of Michael J. Fox, who's done a great service by showing people these so-called levodopa dyskinesias. We've got to get rid of those, because right now levodopa and allied so-called agonist therapies are therapies of choice. So we decided, let's start simple. We are simple. Let's just talk about the problem that a Parkinson's patient has getting going, like starting to walk and stopping. This is something that we all know eventually becomes a real problem. Well, we want to know why this is so tough for the Parkinson's patient. We know that the levodopa, which replaces the dopamine that's gradually going away, we know it helps. We don't understand why. And again, I say, in our lab, in other labs too, we have the belief that this very fundamental research, if we could figure it out, we might be able to help people. So don't laugh. I'm going to tell you about mice. And these mice, we want to see how they do start and stop in a task, such as walking down a corridor and making a decision to turn to the left or turn to the right. And there's a little door in the corridor. That's a little door in this simple psychology T-maze, and we can pull that door away. The animal initiates a movement. He runs down. In our case, we play little musical notes to tell him to go right or left to get to the end of the corridor. And our mice love chocolate, and we give them chocolate at the end of the corridor. And so you can actually see this. I have a couple of videos. Oops, oh no. Yeah. The sound you hear is the sound of neurons. You'll hear them again. Little tiny, quick electrical bits of information. There goes a mouse. You got to the end, you got it. I'm going to show you 3 runs. There he goes. This time the other side was baited. You can't see the baited. Now watch on the 3rd one. Did you see him try to figure out which way to go? I like to think that he's an MIT mouse. So he did figure it out. Now what did we do? Well, you may have noticed that this mouse is carrying a funny looking hat, and this hat actually carries a lot of very, very, very fine wires, maybe 12 microns in diameter, down deep into that pink zone of the mouse, into the striatum. And so all we decided to do is to say, okay, let's take a naive mouse or rat, let's have him learn to do this behavior, and let's see what happens to the neurons in there. It's something that had not been known. And I'm going to show you a color plot to show you what happens. And the first thing to say is, here's the maze, and the maze time, the run times, they go this way. So here's the warning cue, the gate opens, he starts. Here are those tones that tell him to go right or left. He turns, gets done with a turn, and gets to goal. So that's time. And we're going to follow the activity as he learns, and learning is going to go down this way. And the bright red shows you high activity of those nerve cells in the striatum. And so what you can see looks rather, I guess, dull, in that as he's running down, there are tons of neurons that fire a lot over baseline, and they're firing away, and it looks sort of like they just fire a lot. That's it. And this graph stops when he learns. That's the sort of psychology criterion for learning. But look what happens when he really learns that task. We were stunned by this, and this is now, we've seen this a number of times in different species actually, that activity that was all over begins to pile up, guess where? At the beginning and the end of the runs. So we're looking at each other, we're saying, wait a minute. So we actually have spent some years looking at this. No, it's not velocity, it's not acceleration or deceleration, and a number of different things. These seem to be some kind of communication or computational signal, some kind of brain talk. So, of course, we got very excited about this because you saw that these signals weren't there when the mouse didn't know how to do the task. These were learned, acquired signals. The brain learned these signals. And curiously enough, when we learn to do something, like come as a routine to a room like this, I reckon we probably have them too. And then maybe we just package that behavior and say, you know, this is good behavior to have. Why don't I put little brain markers, little signals around it? That's our idea, that somehow action boundary representations form, and we've done a lot of work on this. We know that time is embedded in these representations, but I won't show you all of that. What I will say is that, of course, it came to mind to say, you don't suppose this has anything to do with Parkinson's disease. And I cannot truly say that we know, but there is work that's been done by some people in the room, and also a very wonderful postdoc, Lydia Hernandez, who just went back to Spain about a week ago. And I'm going to tell you what we've been doing to try to find out whether those crazy beginning and end signals have anything to do with Parkinson's disease. And the way Lydia and the group decided to work on this is to, shown here, this is a little cross-section through the mouse brain or rat brain. And the pink region is here, that's a striatum. And this little dark part shows where we made experimentally a lesion of the dopamine system such that we mimicked the dying back of the dopamine fibers that occurs in Parkinson's disease. That is to say, in this little region here, the dopamine is just about gone. And we now have very fancy ways to measure that in these brains. So here's the really great setup. This is a hemi-Parkinsonian model, one-sided Parkinsonian model. The animal is perfectly fine to continue to do, or to learn to do the maze test, because we only took out dopamine in one little part, the sensory motor part. And we put electrodes, these that I showed you before in the little mouse, we put them here inside the place where dopamine was gone, and then we put them in the same place on the other side where everything was normal. And I want to show you the shocking result, and actually some of these results are, I don't know whether you saw it in the room, but some of these results have been so recently analyzed that we're still in a state of shock. But let me show you. Now what I'm showing you is what neurons look like on the control side. This is the good side. We didn't take away the dopamine. Sure enough, another set of animals, in fact they've got this kind of thing going on in their heads, they form the beginning and end pattern. That's fine. Now I'm going to click on what goes on in the depleted side for some of the neurons. There it is. They can't do it. This is one subset of the neurons right there in the pink zone, so to say. They can't do beginning and end. Well, those aren't the only neurons in there. There are other neurons, and we do what's called classification, divide them up into groups best we can. And so we have another group, and now I'm showing you dopamine depleted, the bad side of the brain. These guys on the bad side, they can form a beautiful pattern. Even though the dopamine is very, very low, it's just a hint of dopamine left as Mark Howe sitting over there proved. But look what happens when we give them levodopa. What happens is this stuff goes away and they get a most magnificent big signal right at the time the gate has been opened, the guy's got to go. They fire and look at how strong the firing is here compared to before the therapy when they get to the end. That's interesting. So this is it's an experiment. You take the dopamine away and you do it in an experiment and hey, that's not Parkinson's. What about a mouse model of Parkinson's? And by that I mean, how about have there are many, as I said, genes now being discovered that can predispose people to having Parkinson's disease. Suppose we get a mouse who has one of those genes in him, so he's a kind of Parkinsonian model mouse. What's going on with the cells there? And here's the work of Eric Buger, who's boom, right over there, but I don't want to put the laser light in his eyes. And Eric has done a hero's effort of looking at the very first and still extremely famous Parkinson's mutation, so-called alpha-synuclein. And here is the control side with a lot of activity at start in this subset of cells, and doggone it, the alpha-synuclein mutant just doesn't have enough. So what I'm telling you is that even starting with something so straightforward as just what is it that Parkinson's people really have trouble doing, if we go into the brain and try to record the activity signals that might be related to that behavior, we can find that those signals are very, very elegant. They're very beautifully organized. They develop as we learn, and they definitely are badly affected in at least 2 models of Parkinson's disease, in which the amount of dopamine in the pink zone, the striatum, has gone way down. Can we fix this? Geez, I wish I could, isn't this an opening? Now, the next slide, we fixed it, right? Well, of course, we haven't, but we're trying. So what we have to get at the dopamine-containing neurons themselves. And this sounds easy, but it's not. So, you can't see this. You've got to see this. We have to turn the light down. It's just too pretty. So we should have this thing on. I really want them to see it. This is an IQ test, obviously.
Yeah,
Audience listening. Dim. Oh. Yeah, but it needs to go farther.
I think it will dim slowly.
Oh, it's because of filming. Oh, can you see?
You can see.
Well, just for a second. Just for a second. Because, gosh, never mind. Anyway, there's some red cells there. And those cells, this is what we're after. These are dopamine-containing neurons, and we know because they're red. Isn't it pretty? Was it worth it? So nowadays, very, very recently, partly developed here at MIBR and partly at Stanford, you can engineer, listen to this, a virus to carry the molecule of your choice into the brain. And MJ Kim, whose name is here, has done that in this animal. And these green cells here, they have a very special virus that's in them. And in this, which again I'm afraid is hard for you to see with all these lights, these yellowy things, these are the cells that are bona fide. We can turn the lights back on. Those are, do turn the lights on. Those are dopamine-containing neurons that now have been infected by a virus that has been engineered so it will only infect dopamine-containing neurons. Now, thanks to the wonders of very, very recent of science, including work by Carl Deiseroth and two people here, Feng Zhang and Ed Boyden, we can now make that hat that the mice wear also have a light guide. And here you see this blue light, and this thing sits on, is really here. And so we can put this little light fiber down right to where the virus infected the dopamine cells. And if we turn that light on, we can make the dopamine cells turn on and get active, or we can turn them off. And I thank you, Janet Atkins, for helping us with this. This is a very, very exciting experiment. It hasn't been done before, to our knowledge, and we're going to have animals who are doing a task who will then have us manipulate whether the dopamine is on or off. And we're extremely much hoping that that will give us a whole new view of what might be going on. It's crazy that this isn't known. It just isn't. Now, another reason why it isn't known is that we haven't had a good way to stick a probe down into the pink zone, the striatum, and actually measure how much dopamine is there. We can do it with a very crude method, but the time scale of the method is many minutes. Like something happens in the brain and you don't get your readout until 20 or 30 minutes later. Now there is a way to do this, and this is an engineered job by Mark Howe, who's also sitting over there about to be electrocuted by the laser. And we can put in this same headset that the little animals carry, by the way, that only weighs 2.2 grams, it's pretty light. We can put down a fiber like this, and Mark is able, in the animal who's running around, to measure in real time how much dopamine is being spewed out of those fibers that were coming up that tract into the pink zone, the ones carrying the dopamine. And when Mark does that, oh, I could look at this all night. These now are pseudo-colored signals showing you the dopamine concentration, and actually this black line shows where the warning cue was in the maze, and the animal starts, and bang, there's a big spurt of dopamine. That's a good fact to know. So everywhere we're turning now, we're seeing that probably part of the problem with, that you guys know in the back of the room and some others here, the trouble with getting yourself going, or once you go, stopping yourself from going, partly may lie in disordering of these signals. And the signals are getting fainter, and we need to find out about them. We need to know exactly what the signals are, when they occur, what influences them, and how to restore them, even if we can't restore yet the dopamine itself. So that is a very important thing. Now, I talked a long time. I want to close by coming back to the levodopa therapy. And the terrible thing that with levodopa therapy, levodopa-induced dyskinesias can occur. That means abnormal movements occur. And so Jill Crittenden, whose name is here, Jill and a small team and I, we had actually discovered some genes. We found these genes are probably important for this generation of these bad movements. And here is one gene array result. The more dyskinesia you have, these are dyskinesia scores in animals who have animal dyskinesias, the more here this red gene is being expressed. This is a gene. The more of it you've got, the more dyskinesias you've got. And so Jill started a wonderful experiment. It's still ongoing. She did a mouse marriage. We had mouse marriage in the lab. We all attended. And the mouse that was married, the two mice that were married, were mice that another PD model mouse, it's got a crazy name. They got married to our mouse, which Jill had created, and this mouse lacks, because she engineered it out, this lacks the gene that you get more of this gene when you've got more dyskinesias. So we're hoping that if that gene has something to do with causing more dyskinesias, if we mate a knockout of that gene, that mouse, to a guy who's got Parkinson's disease, maybe we'll have less dyskinesias. So babies are born, and we're going to find out when they grow up, if they are grown up, some of them, do they have fewer dyskinesias when we give them the levodopa therapy? So the last videos, this is one of those PD model mice. He's sitting in a lab beaker, but nothing very interesting. But this is interesting. Now I'm going to show you two mice. This mouse, I guess I can use this. This mouse over here, I'm going to start the video in a minute. He's the PD model mouse. He's been given levodopa therapy. This mouse over here, he's the attempt at reducing the levodopa therapies. Our mouse has been crossed to the PD mouse, and this guy is an offspring. So if you give levodopa to the PD model mouse, he gets terrible dyskinesias. You can see him jumping around. It's really frightful to see. This is mouse levodopa-induced dyskinesias, and they don't have to be in a jar to do it. And here is the same mouse, only now he lacks the gene we discovered and that Jill has worked so hard on. And you can see how much less this guy is moving around compared to that. So, in summary, we think now that there's some potential for doing targeted gene therapy to help with this problem. And of course, the way you'd like to do the therapy is to have some drug that could be given in association with therapeutic drugs that would help to make those levodopa dyskinesias go away. We are so grateful to you and to the Parkinson's organizations. And I know I talked a long time, but I'd like to say that I personally am deeply grateful to people who have worked on this. I've mentioned several of these names to you today. These are people who are really wonderful and really dedicated, and we're all hoping we can help. Thank you.
Good, thank you, Anne. Well, I've learnt something new today, the metaphor of marriage for studying mouse genetics. And it's indeed a wonderful marriage of basic research and its clinical applications. So thank you again. Our final speaker of the evening is Dr. Marie Saint-Hilaire. Dr. Saint-Hilaire is she received her MD from the University of Montreal, and she is now a faculty member at Boston University Medical Center, where she is an associate professor of neurology. She also directs the Center for Parkinson's disease and movement disorders research there. I was just studying her resume, trying to commit it to memory. And she is also the director of Center for Research Excellence that is supported by the American Parkinson's Disease Association, and she is also a member of the Scientific Advisory Board. Just looking at her resume, I have it here if anyone would like to read it. I'm very impressed at the extraordinary range of your research interests and activities, everything from studying testing the efficacy of new therapies to basic studies on the genetics of Parkinson's disease. So we're honored to have you with us tonight, and looking forward to your presentation. Thank you.
Thank you very much for the introduction. Yeah, that's not my presentation. Thank you so much for inviting me. This is a great honor here to be talking here. So I'm going to just give an update of the clinical research in Parkinson. What are the important directions that the clinicians, I'm mainly a clinician, what we think would be important areas for the treatment, the clinical treatment of people who have Parkinson's disease. go. Excellent. Okay, let's advance. All right, so the important areas of research I'm going to talk about today are the preclinical diagnosis, the trying to find biomarkers, neuroprotection, which is an important field of research in clinical research in Parkinson's disease, and we'll talk a little bit about new symptomatic treatment. I just want to show you the difference, how the shift there's been a shift in how we treat Parkinson's disease. So initially, or what we do now, is that we make a diagnosis of Parkinson's disease based on clinic. Somebody presents with the classical symptoms of Parkinson, the tremor, the rigidity, the slowness, and really it's a neurologist who has experience with Parkinson's disease who makes the diagnosis. We, at this point, have no blood tests. It's not an MRI or a PET scan that will help us to make the diagnosis. So by that time, people say about 70% of the dopamine cells have already died in the brain. And the way we treat people is by giving them L-Dopa or Sinemet. It's by replacing dopamine. So there are other medications, dopamine agonists, and MAOB inhibitors and COMT inhibitors. But the goal of all these medications is basically to increase the level of dopamine in the brain. But there are two caveats. One, Dr. Graybiel talked about it, is that the medication causes abnormal involuntary movements called dyskinesia. And the other one is as the disease progresses, patients have symptoms that do not respond to dopamine. So it appears that dopamine is not the only answer. There are other neurotransmitters that may be involved, and we don't replace them well at this point. So there are certain symptoms such as depression, autonomic dysfunction, which includes low blood pressure, severe constipation, urinary problems. Some people develop cognitive problems, and all these problems do not respond well to the levodopa. So now what we want to do is the new target. What we're trying to do in clinical research in Parkinson is do an early diagnosis and try to treat the cause or find a treatment that will either slow down the disease, ideally would stop the disease progression. So we know that Parkinson's disease may start years before the motor symptoms appear. And there are certain things, certain signs or symptoms that seem to be more associated to this prodromal phase, which could be sometimes even 15 years before the motor symptoms start. The one that is best known is olfactory loss. Losing the sense of smell, and some patients will tell us that they've lost their sense of smell years and years and years ago. Another symptom is something called REM behavior disorder. So the person is not paralyzed normally like we should be during the dream. When we dream, we're usually paralyzed, our muscles don't move. But people who have that disorder will not be paralyzed during their dream. Their brain is very active during the dream. So people scream and trash, sometimes fall out of bed when they dream. Other things are autonomic disorders, especially constipation. And some people might have depression or anxiety years before they're diagnosed with their Parkinson's disease. So what we think is that there's this, I'll use that little pointer, prodromal phase. And there's this time of a few years where people do not have the tremor or the rigidity of Parkinson's disease, but have all these other symptoms. And then eventually, when the loss of dopamine gets worse or reaches an area of the brain controlling movement, then they start to develop the motor symptoms of Parkinson. So what we're trying to do is try to find out who's there. And if we have good treatment eventually that is neuroprotective, give the treatment there. That's our goal. So there's this big study that is taking place now, funded by the Department of Defense, called the PARS study. It's to find out if we can identify risk factors or identify people who have the risk factor who may be at risk for developing Parkinson's disease. So it was a multi-center study. We were a center at BU, but of course we had like 15,000 individuals, so there were many centers doing it. So there were people who had Parkinson's disease relatives, or people who were not relatives of Parkinson's disease, but all these people did not have the disease. Okay, they were just volunteer. And to screen all these people, it was screened through the mail. We sent them a special smell test where they scratch and sniff, and they returned the test. A subset of these people came in to be evaluated clinically, and they got imaging. And then about 450 of these subjects will be followed for 5 years with the imaging and the smell tests and cognitive tests and clinical tests. So we will try to see if we can detect in advance, so predict in advance, who is at risk of developing Parkinson. So far what we found is, you'll see the column on the left, so people had the very, very lower sense of smell. So they were below the 11th percentile of normal for their sense of smell. So they had decreased sense of smell, had a much higher risk of having a lot of symptoms I talked about: the constipation, the depression, the anxiety, the REM behavior disorder, of course, was elevated. So if we think 1 is normal, is so anything above 1 is a higher risk. So for REM behavior disorder, it was 2.47. And it was even higher risk if you had 2 or more of these symptoms. So there seems to be a constellation of symptoms that if we follow people, we might be able to see who would be at risk of developing Parkinson. So that's one area of study. The other one that's very important is trying to find a biomarker. As I mentioned in the beginning, the diagnosis of Parkinson is made on a clinical basis by an experienced physician. It's only when the motor symptoms have appeared that we can make the diagnosis. Okay? But we don't have any blood tests. It's not an MRI. It's not a PET scan. It's not even genetic testing that tells us who has Parkinson's disease. So a biomarker is really a sign that helps you diagnose a disease. For example, if you have sugar in your urine, you probably have diabetes. Okay. So these are the potential biomarkers that have been studied. I mentioned the smell test, constipation. This is a PET scan. Genetic testing. Some people have looked at cardiac innervation and at the transcranial ultrasound of the substantia nigra. So the Michael J. Fox Foundation has put a lot of money on trying to find a biomarker and has funded a study called the ppmI study. Basically, it is 600 subjects overall, so 400 will have Parkinson and 200 are controls. And they follow a very strict protocol. All these subjects who have Parkinson's disease are very early on in their disease. They're not taking any medication yet because they want them to be studied before the effect of the medication could interfere with the result of the testing. These they go through a very standardized protocol of evaluation. So this includes mentioned the DAT scan, which is a form of nuclear scan, MRI, motor assessment, neuropsychiatric cognitive testing. They go through several spinal taps, check their urine, store their DNA. So, and eventually, we're trying to find if we can find anything in all this information that we are gathering that would help us to test for developing Parkinson's, for diagnosing Parkinson's disease. This is just to show you a DAT scan or beta CIT scan, which measures, as Dr. Graybiel mentioned, the amount of dopamine in the brain. And you see on the right, it's brighter than on the left. So we know Parkinson's disease also always starts asymmetrically, so it's always worse on one side of the brain than the other. So the left side, which controls the right side of your body, is, you have decreased dopamine, so your Parkinson's is worse on your right side. And you see as the disease progresses, the amount of dopamine decreases in the brain. So this is a test that is used at the moment for research. We use it also sometimes to confirm Parkinsonism, but it's not a test that is useful to differentiate the various forms of Parkinsonism, idiopathic Parkinson's disease versus other forms such as multisystem atrophy of progressive supranuclear palsy. But it does help us to confirm that at least the people who come into the study do have a form of Parkinsonism. So just to a little summary, you see that the PARS, we're trying to find people before they show the motor signs of Parkinson's disease. And the ppmI study, it's when people have already been diagnosed with Parkinson's disease, trying to find a biomarker. And the goal is really, because if we can diagnose it really early or preclinically, is to start a neuroprotective treatment. This is the holy grail of neurology and treating Parkinson's disease. We really want to stop the disease, find a treatment very early on, and then prevent the progression of the disease and prevent these complications and even the motor complications. So this is, you don't have to read all this, but this is just a list of all the medications and treatments that have been tried so far as neuroprotective treatment in Parkinson's. So we haven't found anything very promising yet, but these are all clinical studies. These all studies were done in patients who volunteered their time for science. People have heard about CoQ10. This has been proven now that it is not effective. There are 2 other studies that are going on at the moment, and there are several others. One is creatine, one is inosine. I talked about inosine because it's done across the river here and at Boston University also, where we give a substance. It's a supplement, in fact, not even a medication. It's a supplement that increases the level of uric acid in the blood. And in big epidemiological studies, it has been shown that people who have a higher level of uric acid have a slower rate of progression. So the goal is trying to elevate the uric acid, but keep it within normal range because we don't want to cause gout. But the patients are wonderful. They monitor their urine pH. The way to do it is monitoring urine pH to prevent kidney stones, and they have visits every 2 weeks, and we measure their levels. And the other medications called isradipine. It's an old blood pressure medication, a calcium channel blocker, which has been on the market for many years. It's not used very much, but there are studies showing that that it could be neuroprotective in Parkinson's. So that study is also going on in patients. I want to mention something about exercise because there's a lot of data in animals, and I'm presenting one study. There are many studies in mouse and model and even in monkey models showing that exercise, physical exercise, could have a role in neuroprotection. It's unfortunately very difficult to prove in humans. But if you create a mouse model with giving them a compound called MPTP, and then you exercise those mice, you find that the mice who received MPTP compared to the mice who received MPTP and did not exercise have less impairment in their movements than the mice who did not exercise. And this was associated with some improved brain function and some neurotrophic factors, secretion in the brain. So it's very difficult to prove in humans. You cannot tell a group of people who have Parkinson's disease, okay, you exercise 3 times a week, and you tell another group, no, don't do anything. It's, these studies in neuroprotection for Parkinson have to be done over 3 to 5 years. It's very long range because the loss of dopamine, the progression is slow. So these are very expensive. You require hundreds of people per group. So, I mean, people are trying to design a study to do that, but it has been challenging. We know, at least for epidemiological study, and there are several epidemiological studies, this is also one of them, showing that these were a big cohort of 143,000 people who were followed in a cancer prevention study. And what they found was the people who had the highest category of recreational activity, so people who exercise the most, had a lower risk of developing Parkinson's disease. So either Parkinson's disease reduces your exercise reduces your risk of Parkinson's disease, or maybe if you have preclinical Parkinson's disease, maybe you don't exercise as much. But this is why we always tell our patients who have been newly diagnosed with Parkinson's, it's very important to do exercise very early and put them on an exercise program as soon as they're diagnosed. I'll finish with the symptomatic treatment. There are a lot of treatments that have been developed at the moment. I'll just mention a few in passing. There are some new levodopa formulations, something called duodopa, which is a gel that brings the levodopa directly into the stomach through a G-tube. The problem with Sinemet, the regular Sinemet, is that the half-life is very short. It's 90 minutes. So when a person has advanced Parkinson's disease, they might have to take their medication every 90 minutes to 2 hours because the duration of action is very short in advanced Parkinson. So the duodopa is a way to give the L-dopa, the precursor to dopamine, on a more even, continuous basis. Unfortunately, L-dopa is not very soluble, so you cannot really put it in a patch. It's very difficult to put in a patch, and it cannot be given subcutaneously. Other medications that are being studied, and Dr. Graybiel talked about the problems with dyskinesia, so some antidyskinetic medications are being studied at the moment. Presently, the only thing to do if you have very severe dyskinesia, there's one medication called amantadine, which might help a little bit. Otherwise, it's going to deep brain stimulation. So there are studies being done on rehabilitation and exercise, new adjunctive medications to help the L-dopa or Sinemet work better, some studies on magnetic stimulation, and some surgical therapies. The deep brain stimulation is really not a research surgery, because really it's really approved by the FDA, and a lot of people are going through the surgery when they have advanced Parkinson. But the ones that are being studied at the moment, one is Nurtrin, and that didn't seem to be giving any great result. It's introducing a neurotrophic factors directly into the brain. One is introducing a gene that produce glutamic acid decarboxylase. So basically, they attach the gene to an inactivated virus and put it in the area of the brain called the subthalamic nucleus. And basically, that gene does what the deep brain stimulation does. It's just a chemical deep brain stimulation. And Pro7 is a combination of genes that would be also introduced in the brain and help to increase the level of dopamine in the brain. So these are presented as gene therapy, but they don't really correct the abnormal gene that's causing Parkinson. It's basically a genetic way to either increase the level of dopamine in the brain or to enhance the neurotrophic factors in the brain. There's, at the moment, very little brain stem cell research being done clinically in patients with Parkinson. I know people are always interested about stem cells. At the moment, stem cells is still mostly studied in animals. There's one study of autologous bone marrow derived stem cells where they took the own the patient's own stem cells and implanted them in the basal ganglia of somebody who had Parkinson's disease, and they got very good results, but that was on very few patients, and there was no control group, and we always like to have a control group. The one that seemed to be promising is that glutamic acid decarboxylase therapy. So it is basically introducing a gene in the subthalamic nucleus that sort of decreases the activity of that nucleus. And it does the same thing as deep brain stimulation does, but without having all the hardware that people have when they have deep brain stimulation, which includes having an electrode in the brain, a wire that goes under the skin, and a battery under the skin, and the battery has to be, how can I say, change from time to time. So there's a lot of hardware. It can be infected, it can break. This is basically doing the same thing as the brain stimulation, but just by putting in a gene that would do the same thing, decrease the activity of that nucleus. And that seems very promising, and there will be other studies about that. That's it.
Great. Well, thank you, Marie, and thanks again to all of our speakers. I'd like to suggest that the speakers all come up and sit at the front. We have 3 speakers and 3 chairs, so that way you will be able to answer questions. I think Bob Decimer, Bob is happy to apparently let me moderate. So in the hot seat. So questions for our speakers? Yes. I see.
As our physicians from Montreal, I understand the French-Canadian gene pool is rather limited, as only several thousand people settled up there initially, and several, I guess 70,000 were up there when the British took over. Has that area been a hotbed of genetic problems?
Yes, in Quebec, there are certain conditions that have been described there. For example, at Taxi de Charleroi-Saguenay, and there's the incidence of Tay-Sachs disease in certain areas of Quebec is even higher than in the Ashkenazi Jewish population. Yeah. So there are several diseases that are more frequent in that area.
I know my gene pool keeps running across relatives in it. They kept marrying their cousins.
Good. Other questions? Yes.
If I were looking for an early marker,
Do you want to?
if I were looking for an early, no. Let me try to get rid of it.
Yeah, I think if you speak loudly.
Yeah.
I'll try to speak. You can repeat my question. If I were looking for a marker, I'd look for behavioral markers. Anybody looking for behavior, like people with Parkinson's don't wave their arms and they have a certain gait. I'd look for what they were doing rather than what was happening in their body.
That's the brightest.
Okay, the question is looking for behavior markers. Well, what they're doing is really, it's movement, it's motor, whether it's...
Right, I'd look for movements rather than things going on in the brain.
Well, by that time, it means that the disease has advanced to the point where you see the motor aspect. We want to see it before the motor aspect. And there are people who are doing a lot of research, I didn't talk about that, about cognition in Parkinson's disease. There's a lot of research being done, maybe some of it even here with functional MRIs and PET scans.
Good. Jim has a
question, and that is, if I understood him correctly last night, I believe Ulla Isaacson was doing some research at McLean using pluripotent stem cells through the Janisch lab, and seemed to have extremely good results. So it's only 5 or 6 patients, but they were getting good results over, 6 years? Did I understand that correctly?
I don't know where they do patients. They do patients?
On people, on patients, yes.
I don't know where they do patients.
Only is, well, you probably are giving us the information, Jim.
Yeah.
You were there too, Pat. Is that?
That's what I heard.
I wasn't aware that he was doing it on patients.
Okay.
That's a question.
excuse me, does gender appear to play a role in either the incidence of Parkinson's or in its progression?
Yes, Parkinson is more frequent in men than in women. We don't know why. It's not very well understood. They look at estrogens and hormones, but it's not very well understood. Dr. Graybiel will have another, might know more about that. As far as progression is concerned, we know that women tend to have more dyskinesias than men, clinically. We know also that men appear to have more cognitive problems than women. Why there's a gender difference?
Just it's a wonderful question. There are a lot of studies now on that very issue, many of which we talked about, and there are some signs that the dopamine-containing neurons themselves may differ in men and women.
Yes, Pat.
Can you say something? Can you comment on whether there's any familial connections, and if there are, do you start looking at family members early on?
Very good question. Sorry about this. It appears that if you have a member of your family who has Parkinson's disease, you might be at slightly increased risk of having Parkinson. There are certain genes that have been described that cause Parkinson's. Some of them are very clear that they cause Parkinson. They're like autosomal dominant. If one of your parents has it, you have a 50-50 chance of getting Parkinson. Is very, very rare. It's that synuclein gene, extremely rare, found only in a few families. There are a certain gene called LARC2, which is probably more frequent, that is affecting mostly people around the Mediterranean and Scandinavian Jews, that really increases very much your risk of having Parkinson's disease, but it doesn't mean that you're going to have Parkinson's disease, it just means that your risk is very increased. We have a very big genetic study of families who have Parkinson's disease at BU. And most of the people in that study, so these are all people who have more than 2 members of the family who have Parkinson's disease, most of these people do not have any of the genes that have been so far discovered or described. So there are many genes that have not been found, or maybe it's not one gene, it's a combination of genes that would cause the increased risk of Parkinson. So at this point, we don't use genetic testing unless there's a very specific question to make the diagnosis.
Environmental. You want to talk about environmental?
There may be some things in the environment which could at least increase your risk of Parkinson's disease. And some people think that maybe if you have a gene predisposing you with the right environment, that could cause a Parkinson. This was, it was proposed by Dr. Barbeau, André Barbeau, in fact in Quebec, showing that there was an increased risk of developing Parkinson if you lived in certain rural areas where they spread pesticide or near paper mills. So that has led to another area of research of environmental factors that cause Parkinson's. For example, rotenone, that is a pesticide, is used as a model of Parkinson's disease in animals. So maybe it's not a gene by itself, but a combination of a gene and the right environment that could cause, or wrong environment that could cause the Parkinson.
I think maybe just a couple more questions, because we do have a reception outside, and so we can continue the conversation informally. But I think 2 people have had their hand up, you, madam, and then the gentleman behind, the lady in the green jacket.
I wondered if there's been any much work done on interactions of the work you've done with behavioral or emotional factors in individuals, the increased number of placebo impact, for example, or you talked about chocolate being the motivator. Maybe if you used ice cream, it would create a different impact. I'm just being facetious, but.
I'll defer to our physician panelists, but I will say that it's stunning what this so-called placebo effect does. The scans that you showed, and I did too, are PET scans of dopamine. Dopamine can be increased enormously by such things as chocolate, and by the very thought that maybe you'd get better if you take a certain therapy. So this is good, and it speaks to something. I think we would all agree that to quite a level, this is a situational kind of effect. In other words, in many of these disorders that affect the striatum and the basal ganglia, the symptoms can be worse in some situations. For example, anxiety can bring out problems. Again, I turn to Michael J. Fox, who has these terrible dyskinesias, and he makes a very strong point about it. For example, in his interview with Katie Couric, that, when he's under pressure, for example, in such an interview with a television camera staring right at him, that his dyskinesias are ever so much worse than otherwise. And you just have to think, and we now in science do think, that state, so it's now the jargon is to call that state, that some state of mind, some modulators in the brain can influence what otherwise you would think is a more steady situation. It's a wonderful question.
So I know we had one more question, sir. If you wanted to ask your question before we wrap up, I know you had your hand up.
Mine was answered.
Okay, so we've answered everybody's questions. That's wonderful. So good, thank you, and thank you again to all of our speakers.